Okay, so to define a sample, I have to backtrack and first define a biospecimen. A biospecimen is the primary biological material collected from a patient or participant, and a sample can either be that primary material or a derivative made from the biospecimen.
My working definition of a sample or a biosample is a biomaterial that anybody would want to investigate, ask a question about, or confirm any kind of a biological underpinning at any time now or in the future.
A sample is typically one type. Either it's DNA, it's blood, it's RNA, and we can make multiple derivatives of it, and then we can split it into vials or fragments. But I think the sample is that original donation from the patient.
I think from a biologic point of view, a sample is could be any body fluid, blood, urine, DNA that comes at a given time point from a patient.
A sample for us is either a biological or a chemical collection or substance that is stored for future purpose in research or analysis of properties of these samples.
Yeah, I think a sample for us is an aliquot or a piece of a patient's or human biospecimen that most importantly reveals the molecular realities as if the sample is still in the human body. These samples are used in translational research, drug discovery programs, companion diagnostic development programs too, validation of targets, of drug targets, of biomarkers. You do antibody validation, you do of course all of the gene expression analysis, protein profiling.
So I'll give you one very exciting example that's happening at Duke, and that's in the brain tumor immunotherapy program. They use this material to derive antibodies against the brain tumor for an individual patient. So it's an individual treatment for each patient, and they take that material back into the research lab and a manufacturing facility, create this vaccine against the brain tumor, and bring it back into the patient as part of a clinical trial.
These samples, or some samples, are so hard to come by. It's critical that we really maximize these biological resources so that we can not only create discoveries today, but validate those findings in the future, which will ultimately help develop therapeutic regimens, diagnostics, and really bring the personalized medicine arena to reality.
The biggest driver, at least within our consortium, is science. If there is a possibility that a science will help with identifying a high-risk group of population or will tell us a therapeutic response with a drug or early diagnosis, we would collaborate very heavily.
Bioresources and biospecimens are very, very precious. They come from patients, they're a donation, they're a gift.